All right, it's my pleasure to introduce Dr. Yoandy Coca. He's a chemical engineer with a PhD in chemical engineering and bioprocesses from the Pontifical Catholic University of Chile. His research has focused on the microbiome variation in Atlantic salmon in both open net pens and wrasse, with an emphasis on infections caused by Pischarickettsia salmonis, primarily in southern Chile. Looking forward to your presentation.
Okay, thank you so much, Dr. Professor. So, yeah, my name is Yoandy Coca and I am postdoc fellow at Aquatic Animal Health Lab at UC Davis under Esteban Soro supervision. And today I want to share with you our latest finding in susceptibility of Nile tilapia fingerlings to different Lactococcus petauri genotype isolate in the United States. And with that in mind, let's jump right in. So first of all, I want to start pointing out that tilapia is a generic name for a group of African fish of the family Cichlid. So there are many different species of tilapia, but amongst the most popular one and used in aquaculture, we have blue tilapia, Mozambique tilapia, and Nile tilapia. And I will dare to say that Nile tilapia is probably one of the more worldwide spread and used in aquaculture. And specifically about Nile tilapia, this report for 2022 of the FAO ranked tilapia as number 4 of the aquatic animal produced for aquaculture with 5.3 million tons, which is like a really high amount of tilapia. And the major tilapia producers worldwide, we have those countries with tropical weather because, like our friend here, Ernest Hemingway, tilapia thrive in tropical weathers. And we have different countries that produce tilapia, like Costa Rica, Honduras, Dominican Republic, and also some southern countries like Brazil, Colombia, Ecuador. Also, many African countries like Zambia, Mozambique, Nigeria, and South Africa also produce tilapia. But without any doubt, China is one of the largest tilapia producers. So what happened here in the United States? So according with this census of 2023, for this specific year, United States reported has like 147 tilapia farm, and as you can see in the map, they are spread all over the country. And it's fair to say that some states have more farm than others, like for example Florida and Ohio and Hawaii. But other states also have important number of farms, like California and Texas. But what is interesting about this data is that the amount of tilapia that is produced here in the state is not enough to cover the market demand, the domestic market demand. And this another report from FAO of 2025 highlight that the amount of tilapias imported for 2024 was 67,000 ton of tilapia, and this doesn't include like the whole tilapia. This is only fillet and or other form of tilapia like shield tilapia that came from closer countries like Brazil or Colombia. So, uh, if you so moving on to some challenge of the tilapia production, if you ask people that are not really involved in tilapia production, this question, are tilapias pathogen free? Many people will answer yes, tilapia, nothing can kill tilapia, but this is not true. So tilapias are susceptible to a different kinds of disease like fungal disease, viruses disease, parasite, and of course bacterial diseases. And about specific bacterial diseases in tilapia aquaculture, I want to do some go a little deeper because in this moment we can say that we have four current bacterial diseases of significant importance that might affect tilapia's aquaculture, like Streptococcosis, Aeromoniasis, Franciscellosis, and Vibrosis. And also we have some emerging bacterial disease of concern like Edwardscellosis and Lactococcosis. And about Lactococcosis, I want to go a little deeper, specifically in this disease for the importance that it has for the state of California. So, lactococcosis is a systemic disease that has been reported can be produced for three different pathogens like Lactococcus garbae, Lactococcus firmicensis, and Lactococcus petrauri. And specifically, Lactococcus petrauri has been like a recent concern in many countries, in nations that produce tilapia like Brazil and Zambia, and also here in the state of California, Lactococcus peteuri has become like a increasing problem in farm of rainbow trout. And for this reason, our lab has been working like really hard in trying to figure it out the virulence and potential risks of this specific pathogen. And about this, as I was saying, our lab in 2023 carrying out this really interesting research for Eric Litterman, in which they challenged Nile tilapia uh, with the pathogen Lactococcus peteuri at 18 degrees Celsius, and they didn't find nothing really alarming about this challenge when they infect the tilapias with the pathogen at 18 degrees Celsius. However, it's fair to say that this particular research highlight that the virulence of Lactococcus peteuri is really linked with temperature, and for that reason, we decide to keep exploring and going deeper in this area and explore two different and higher temperature. And also, we decide to explore two different genotypes of Lactococcus peteuri for two isolate for two different hosts, one for hybrid catfish, another for rainbow trout here in California, because also these two species are have a really are really important here in the United States, and we and also because of course California produces like a significant amount of trout. So with this background in mind, our main research question was: Is Lactococcus peteuri virulence in night tilapia influenced by strain genotype and temperature? And for answer this question, we implement the following experimental design in which we those two Lactococcus peteris genotypes I was talking about, and we challenge or tilapia intracelomic for intracelomic injection, and also with immersion for 1 hour. And we set two bank of tanks at two different temperatures, so 30 degrees Celsius and 25 degrees Celsius. And we track the experiment for 2 weeks and a half, around 17 days, and we collect water weekly, and also we track daily mortalities of the challenge, and also we play the brain of those mortalities for in agar blood play to recover the pathogen. And also at the end of the challenge, we took some fish, three fish for challenge, and for histological analysis, and we also played the brain of all survival fish. So for the qPCR quantification, we use the standard curve that was developed for Dr. Taylor Herkman in his work redefining patient lactococcosis, and we consider as positive those samples with CT values below 35. So, moving on to our results, here is what we found about this challenge. So, first of all, about the detection of Lactococcus peteris in water, we indeed detect um, Lactococcus 6 hours and 1 day after our challenge at both temperature. However, and in all uh, the challenge on intracolonic injection and immersion. However, this was expected for because obviously the specifically in that immersion challenge, the bacteria was in the skin of the fish, and this leaking process was expected. But what's really interesting is that we didn't detect in the following samplings any Lactococcus peteris in the system, which basically means that the tilapias were not sharing the pathogen in the environment and into the water. And this is consistent with our next result, and is that we got 100% of survivability in those fish that we challenged at 20 that was exposed at 25 degrees Celsius by intracelomic injection and also immersion, and we were only capable of to recover, and we only detect one fish that died potentially for Lactococcus peteris. So only one fish we are able to detect Lactococcus peteris in the brain. So if we also with really high survivability rates, and if we compare our results with other works that have been carried out, by example, in Brazil for Dr. Renata Edgar, in which she challenged Lactococcus tilapia's fingerlings with one with Lactococcus petauri at 20 degrees Celsius. That is a temperature that is in the middle of the temperature that we explore. And she used in her experiment like lower concentration than us. We use 10 to the 7, and she used approximately 10 to the 6. And she got mortality about 90%, which is really surprising, contrast with our results. So could be that many factors for her experiment, but for example, we are not sure what kind of Lactococcus pituitary genotype she could use in her experiment. So that could be a really important factor to have keep in mind for this difference of between our results. Um, from the histological point of view, the general findings were really similar among the challenge phase and control group without any outstanding histological surprise. So basically, both challenge and control group have presence of pigmented macrophage aggregates and necrofocal cyanosis, but beyond this, nothing really alarming from the histology. Um, with all this information, our overall conclusion is that our data provide strong evidence that the Lactococcus petuaris genotype that we tested here in our lab, the genotype from a rainbow trout and the one from hybrid catfish, have low virulence in Nile tilapia fingerling, as reflected for the absence of clinical signs, and we didn't detect any of the reported clinical signs like a popping eye or like erratic swimming or nothing like that. And also the lack of consistent of the recovery of Lactococcus petuari. Just one fish was detected with the pathogen. Um, which is like basically means that these genotypes that we tested are not virulent for Nile tilapia. So with all this, I want also especially acknowledge the founding sources of this work, the National Institute of Food and Agriculture, for this special grant in aquaculture that allows us to carry out our research, and also all the aquatic health lab members. And with all this, I want to thank you for listening, and I'm more than welcome to take any question that you might have, and I will try to answer them. So that is it.
Great. Thank you very much. Very well done. Folks, if you've got questions, feel free to drop them into the chat, or you can unmute yourself now and ask Yoandy directly. No, you're getting virtual applause. Thank you very much. Great job. Well done.
Thank you, sir.